Okay, in this chapter, we're going to talk about leaves. So leaves are actually similar to solar powers in that they're covered with a transparent epidermis that emits light into the interior, and many twist on their petioles so that their surfaces are inclined to the sun throughout the day. So all leaves originate as primordia. Most leaves consist of a blade, a petiole that may have paired stipules at the base. You notice I said may have. And leaves may be simple or compound, and all are associated with leaf gaps. Okay, the picture on the left is a picture we've looked at many times when we're looking at the meristematic tissue. So you see the leaf primordia that the leaves originate from, where that comes up from the apical meristem, and it's also over there adjacent to the axillary buds or meristem over there on the right and the left. And then on the right side of your screen is just the different parts of the blade of the leaf, which is the apex or the tip, the margin, which are the edges, the veins, the midrib that goes up the center, and the base is at the very bottom of that midrib where the leaf ends. Then you have the petiole, which is what the midrib goes down into, and the stipules are the little things that look kind of like little baby leaves down at the bottom. Okay, so the lower and often the upper surfaces of leaves have pores or stomata that permit air circulation and facilitate photosynthesis. Leaves also transpire. They accumulate waste and eliminate excess moisture via transpiration. And the sizes and shapes and textures of leaves vary greatly. So the picture over here, the two little things that look like this, those are your stomata. We actually saw some of those when we did some of the plant slides that we made ourselves. Okay, this is just a reminder on photosynthesis and how this happens in the leaf. The water arises from the roots and exits through the stomata. So it would actually come in through the stem. You've got the sunlight coming in. You've got the CO2 that's coming in, usually at the bottom of the leaves. And then you have transpiration, which is the moisture also going out, as well as O2, which is oxygen. Okay, and if you're going, what is transpiration? Well, transpiration is just when water evaporates from plants. That's just what the process is called. So plants lose water through their stems, leaves, and roots. And a fully grown tree may lose several hundred gallons of water through its leaves on a hot day. Sometimes you'll even see a plant that all of the water is actually, you can see it just balled up on the very edges of the leaf. Sometimes it's all over. All right, the veination and compounding of leaves may be either pinnate or palmate. And the main vein is the midvein in simple leaves. And the rachis is what it is called in compound leaves. So monocot leaves, we now have parallel veination, where dichotomous veins or dicots occur more in a branched fashion. So let's see. The first picture in black that you see here, this is showing palmately compound. So just think about the palm of your hand and how they all arise from one spot, and they are all coming together right there in the middle. And the pinnately ones, and you notice that it says that's a rachis through the middle of it instead of a midvein. The leaflets arrive at several locations along the axis. So you can see the little nodes for the leaves on here. The picture on the right, that's a simple leaf, A is. So that just shows the midvein through the middle. The petiole is the stem where the leaf cuts off that comes down. If you look at the little box that they pulled out, whenever you have a leaf that comes down and then lobes up like this, that little indentation right there in the middle, and leaves have many, many different shapes. That's called a sinus. And then you have the lobe or the tooth. And then once you have your midvein, you also have what we call secondary veins that branch out. Okay, B is going to be a compound, and you can see that it has the petiole, and then each individual leaflet has a petiole that comes in and attaches to the rachis, and then you'll have a terminal leaflet up on the very top. Some of these were kind of hard to tell when I had you guys bring in pictures at labs, because it would have been easier when we go to looking at some of these other slides if we had seen maybe more of the branch that the leaves came off to figure out some of this. Okay, so here's another one to help with this. Up on the top left, you've got a simple leaf, and that's off of a cottonwood. Okay, down below it, the clover, that's a palmately compound leaf. And you notice how they all come in palmately, just like the hand, except there's 3 of them instead of 5. And then you have a pinnately compound leaf, the black walnut, with the rachis right up the middle. And then next is the double compound leaf of the honey locust, and that's the one that we saw when we were doing our little outside field trip that have the really long thorns. But notice on this one, it's double compound because you don't just have a single leaf on each side, you have more petioles and more leaves up each one of them. Okay, so a simple leaf. Let's look at this arrangement here. Okay, you can see the stem and you can see the auxiliary buds really good, and that's marked in red for you there. So we can tell that these 2 leaves are opposite because they're on opposite sides of the leaf stalk. And then if you go and you look at the compound leaf, notice here's the auxiliary bud. This is what we couldn't see on a lot of the samples that you brought in, where the auxiliary bud was. We could see nodes on the rachis, but we couldn't see the auxiliary buds because we didn't have enough of the specimen to look at. So you can see the auxiliary buds, and then now you know that you see all of this, that these are leaflets and that this is a compound leaf. If it were a simple leaf, there would be an auxiliary bud at each leaf, just like in the first picture. Okay, monocot versus dicot leaves. Very simple, and there'll be many, many different names where they will name the veination on your dicot leaves, but on the left is your monocot, so you have the parallel, the veins just run parallel, straight lines, up and down. Then your dicot leaves, they will be branched. And like I said, there's probably a dozen different words if you want to get really specific about the branching. Okay, the epidermis is a surface layer of cells that's coated with a cuticle. And we know we're talking about the leaf. So the waxes, the glands, hairs, and an occasional cellular crystal may also be present. And the lower epidermis usually contains numerous stomata. Each is formed by a pair of guard cells. And we had talked about this before. Those guard cells regulate both evaporation of water from the leaf and the gas exchange between the interior and the outside atmosphere. So you can see on here where the guard cells are at the very bottom, and the stomata is that opening there, and that is where that exchange takes place. So this is the same cross section of a leaf. So the mesophyll between the upper epidermis and the lower epidermis may be divided into an upper palisade layer, which consists of rows of parenchyma cells containing numerous chloroplasts and a lower spongy layer in which the parenchyma cells are loosely arranged. So the veins transverse the mesophyll. Okay, so we'll see the very top is the upper epidermis. The next layer is the palisade mesophyll cells. Then it goes down into the spongy mesophyll cells, and you see lots of airspace and even your vascular bundles in there with your xylem and your phloem. And then, excuse me, the very bottom, your lower epidermis, you can see your guard cells. And then that opening is the stomata. Okay, so this will look more familiar, like something we looked at underneath the microscope. This is your monocot versus your dicot leaf cross section. So if you look at the top one and see you've got that little area that we called like little monkey faces, but notice where the vascular bundles are. They are in parallel to each other, like in a straight row. So that's going to be your monocot on the top. And then on the bottom, this is going to be a cross section of a dicot leaf. So you can see how it's really easy to tell the difference of those. Leaves are a source of many useful products, including, we talked about things like food, beverages, dyes, fuels, spices, flavorings, oils, and they can also be used for medicinal reasons, and they can be narcotics. So what are specialized leaves? Leaves may be specialized usually in adaptation to specific combinations of environmental factors. So specialization includes things like really thick leaves, fewer stomata for water storage levels, tendrils, spines, food storage leaves, flower pot leaves, window leaves, reproductive leaves, bracts, which are just a lot of times are colored, and insect trapping leaves like the Venus flytrap. So such specialized leaves include your protective leaves, your storage leaves, your tendrils, your bracts, insect capturing leaves, and the protective leaves include things like bud scales, prickles, and spines. Okay, I just wanted to show you some that you probably hadn't thought about, but the picture on the left is a beautiful dogwood, and we all want to think of that whole thing as being this really pretty flower. In effect, just that very center part is the flower. The white that we think of as being the petals of the flower are really just colored bracts, and botanically, the entire flower is just that middle section. So these are colored bracts. Your poinsettia, just that part in the middle, the very middle is your flower. All of what you see there, the red leaves, those are bracts. Those are just colored bracts, but they're beautiful. So we think of them as flowers, but botanically the flower is just that very center part. Okay, leaves change color as green chloroplast pigments break down, revealing pigments of other colors. And different pigments accumulate in cell vacuoles. And environmental factors and hormonal changes that occur in autumn in the abscission zone at the petiolar, base of each deciduous leaf causes leaves to drop. Now, I said deciduous because we know that our gymnosperms are not going to drop their leaves. They're what we call our evergreens. So on this diagram, you can actually see the palisade cells of the leaf, the upper surface crowded on the end that receive direct rays from the sunlight. And then as you go down further in there, you've got the minute structures called the plastids that contain the chlorophyll and the green. And the pore, the stoma or the pore down there on the bottom allows the movement we talked about of air into the leaf, CO2 out with the atmosphere, and the loosely packed cells of the spongy layer permit air containing carbon dioxide, which we use for photosynthesis, to move inside the leaf because it's got to have some space in there. And your keratinid pigments are your yellows and oranges that are also found in the plastids. And your anthocyanin pigments are your reds and purples that occur in the sap of the cells. And there is also a transparent waxy layer that emits sunlight while preventing evaporation of water from the leaf cells down there on the bottom, which of course the stomata will come into play there too for the openings. Okie doke. And Stern's introductory plant biology is our reference.